Hi, so in this video we'll talk about the different organs in eukaryotic cells, or organelles rather, in eukaryotic cells, what their basic structure and function is, and then also a little bit about the origin of eukaryotes, and specifically the organelles that are thought to have been originally free-living bacteria. We'll talk about that. Okay, so the nucleus, the defining characteristic of the eukaryotes. All eukaryotes have a nucleus. The nucleus is, one of the definitions of a nucleus is it has to be membrane bound. So this is what's lacking in prokaryotes. Prokaryotes have chromosomes, but they do not have a nucleus because they don't have a membrane bound structure that encloses the chromosomes. Okay, so the nuclear membrane is actually a double membrane. So a membrane is a phospholipid bilayer. The nucleus has two phospholipid bilayers surrounding it. So for something to get across this nuclear membrane, it has to cross the fatty region of one membrane. Then there's a hydrophilic watery area in between. Then there's another fatty membrane that it has to be able to cross. So this is going to make it extremely difficult for any molecule to diffuse across, because if it's hydrophilic, and even if it manages to get across the first membrane, it's going to get stuck in the hydrophilic region in between. If it's hydrophobic, it's going to get stuck in the membrane and it's not going to be able to diffuse across into the hydrophilic region on the other side. So that's what the nuclear pores are for. The nuclear pores are huge protein complexes that regulate the traffic in and out of the nucleus. They allow RNAs to pass back and forth. They allow ribosomes to get out of the nucleus when they are completed. They allow various signaling molecules to get in and out, but they don't allow DNA in or out of the nucleus. This region inside the nucleus, which if we could look through the microscope in lab this semester, we would see is an obviously darker region inside the nucleus that's called the nucleolus. Nucleolus literally means little nucleus in Latin. This is not a membrane-bound region. The nucleolus looks dark because there's a lot of nucleic acid there, specifically RNA. The nucleolus is a site of massive RNA synthesis, as the genes necessary for making ribosomes are being transcribed. So with the various cellular stains that we use to stain cells so we can see structures, these stains stick to negatively charged things like DNA and RNA. So the nucleolus stains darkly just because it contains a lot of RNA, which attracts the dyes that we use. But this is a region where massive amounts of RNA are being made, and that RNA is RNA that the ribosomes are made out of. The ribosomes, as we'll find out later in the semester, ribosomes are mostly made out of RNA, and that's where it is made. In the nucleolus because of the genes that are being transcribed there. The chromosomes are not just DNA. The chromosomes are in fact made up of more than 50% of the mass of the chromosomes is in fact protein. The protein keeps the DNA organized. The DNA that is in our chromosomes is extremely long, and we have many of them in a single nucleus. So the proteins that are associated with DNA, the DNA is wound around the proteins. Proteins keep it organized so that it doesn't get these huge long strands of DNA do not get tangled. So that's what a chromosome is. A chromosome isn't just DNA. A chromosome is DNA plus protein. And we call that material that the chromosomes are made out of. And what we can see in the nucleus here, this kind of darkly stained stuff inside the nucleus, we call that chromatin. That's DNA plus all of the proteins that are associated with it. And in a nucleus like this that's not undergoing cell division, that would also include a lot of RNA as well, as all the genes are being transcribed. The word itself, the root here, chromo, in chromosomes or chromatin, is from the Greek word for color, and that's because early microscopists noticed that whatever cellular stains they used, the nucleus stained darkly. So they called the material inside the nucleus chromatin because it was stained darkly with the stains that they used, and so it had color. So that's where we get that word. So chromosomes are made out of chromatin, which is DNA and the proteins that keep it organized. So here's a nice scanning electron microscope image of a nucleus. So this round thing here, that's the nucleus. And the way they made this image was they froze cells. I don't know what kind of cells these are that they made this image from. And then they roughly cracked open the specimen so that things would tear apart. And then they coated it with metal and bounced the electrons off of it for a scanning electron microscope image. So what we can see here is a three-dimensional surface of the nucleus where part of the nuclear envelope has been torn away. You can see this kind of tear here and here. So what we can see over here is the outside of the outer nuclear membrane. Remember, a nuclear envelope is two membranes. So here that is the outer membrane. We can see these little bumps here. Those are the nuclear pores. And then this inner surface here, this is where the outer membrane was ripped away, and we can see the torn edge here. And then in some places we can see that nuclear pore complex. It looks like a bump. And then in other places, the pore has been torn away. That large protein complex has been torn away. And all we see is holes where it was, holes in the membrane. So this is kind of a cool image of a nucleus for that reason. So now here's a look at the nuclear membrane with the other kind of electron microscope, a transmission electron microscope. So what we see here is the inside of the nucleus. The purple is the membrane, and remember, because it's an electron microscope image, it was colorized. They all start out black and white. And then here's the cytoplasm out here, and some endoplasmic reticulum has been highlighted with the pink and the yellow. But the purple is that double nuclear membrane there. And we can see some pore complexes here with some darker matter going through the pore complex. And then inside here in the nucleus, we can see some of the chromatin. Some of it's looser and less dense, some of it is more dense. So this is what we're looking at there, this double membrane, that very effective barrier. And why did the nuclear membrane evolve? We'll talk more about this when we talk about the evolution of eukaryotes. Ribosomes make proteins. They assemble our completed peptides, proteins from the amino acids. And we'll talk more about exactly how they do this later in the semester. But here's, ribosomes are very, very small. So here is 0.25 micrometers is our scale bar here. So you can see that a single ribosome is much smaller than 1 micrometer. It's smaller than 400 nanometers, which is the limit of what we can see with a light microscope. So that's why we can't see these very well. This is an electron microscope image. With the very best light microscope, at best, the ribosomes might look like tiny, tiny dots, if they can be seen at all. So ribosomes are large molecules. They don't have a membrane. They are made of mostly RNA with some protein that kind of holds them together. They have a large and a small subunit, and their function is to assemble proteins. Now, some proteins are made in the cytoplasm for the use of the cell, and so the free ribosomes do that. So it says here, free ribosomes in the cytosol. So they're just kind of floating around. Many proteins are going to be secreted from the cell and end up outside the cell. Many kinds of proteins are going to end up outside the cell. those proteins are secreted right into a membrane-bound structure called the endoplasmic reticulum, which we'll talk about. The ribosomes stick right to the membrane of the endoplasmic reticulum and make their proteins right into the cavity here in the endoplasmic reticulum. Now those proteins are ones that their destination is eventually to be secreted from the cell, like perhaps peptide hormones like insulin. They're going to be made by ribosomes because it's a peptide, and they're going to be secreted right into the cavity of the endoplasmic reticulum because eventually they're going to end up on the outside of the cell, and we'll talk about how that happens. So all of these many membranes inside of a eukaryotic cell, they're all connected in one way or another. They're either directly connected to each other, or they're connected by vesicle traffic. So when you hear that word vesicle in a eukaryotic cell, that's a membrane-bound structure. And generally vesicles originate with the endoplasmic reticulum. So the endomembrane system includes the nucleus membrane, which is connected to the endoplasmic reticulum. We have smooth and rough, and again here we see things were named by early microscopists based on how they looked because they didn't know the function. So the rough was called rough because it had ribosomes on it and it looked bumpy. The rough is where proteins are being made that are going to be secreted from the cell. The smooth endoplasmic reticulum, it looks smooth because there's no ribosomes on it. Now, reticulum, this word reticulum, the word reticulated means striped. So you'll see in many of the microscope images that I'm going to show you, the endoplasmic reticulum looks very striped. Even though it's not really striped, it's more like a series of pancakes that are all layered on top of each other. But to the microscopist, it looked very stripy or reticulated. So that's why we call it the endoplasmic reticulum. Endoplasmic meaning that it's a membrane inside the cell that looks stripy, and sometimes it's rough and covered with dots, and sometimes it's smooth. So that was how it was originally named. Now we know what it does. Now we know it's the manufacturing center for the cell. Synthesis of all kinds of stuff, carbohydrates, proteins if they're being secreted from the cell, fatty acids, these are nucleotides, these are made in the endoplasmic reticulum. So to export them from the cell, there's another membrane-bound structure called the Golgi, which is usually as well further away from the nucleus than the endoplasmic reticulum is. And we'll talk more about both of those things. So these things are included in the endomembrane system, the nuclear envelope, which is directly connected to the endoplasmic reticulum. Endoplasmic reticulum makes vesicles. Some of those vesicles are going to be lysosomes and also peroxisomes, which we'll talk about. Some of those vesicles go to the Golgi apparatus if things are going to be secreted from the cell. Some of those vesicles connect to vacuoles, like in plants. They have a large central vacuole that is connected to the endoplasmic reticulum by vesicle traffic. And then the outer plasma membrane of the cell, the outer plasma membrane that surrounds the eukaryotic cell, is constantly being refreshed by new membrane that's made in the smooth endoplasmic reticulum. So they're all connected. All the membranes in a cell are all connected either continuously or by vesicle traffic. So when we say vesicle for a eukaryotic cell, it has a membrane. It's surrounded by a membrane. So here's another nice TEM image of smooth and rough endoplasmic reticulum. You can see how it looks kind of like it's striped. But we now know that these stripes are actually thin slices through larger kind of pancake-shaped structures that overlap each other and are connected with each other. So the smooth endoplasmic reticulum, this is for synthesizing pretty much everything that's not a protein. So synthesis of lipids, phospholipids, steroid hormones, It's also used in specialized cells like in the liver for detoxification. And it's used in your muscles for muscle contraction. So calcium is released from the endoplasmic reticulum when your muscles contract to trigger muscle contraction. The rough endoplasmic reticulum, it's rough because of ribosomes, so rough ribosomes. Ribosomes make protein. The rough endoplasmic reticulum then is all about protein synthesis, specifically things that are going to be secreted from the cell, and also proteins that are going to stick in the membrane on the outside of the cell. So those proteins are made, secreted right into the membrane that's going to eventually end up on the outside of the cell. So the Golgi apparatus, this is, if the endoplasmic reticulum is the factory, the Golgi is the FedEx of the cell. So vesicles, this is a nice TEM of a Golgi. So, and the Golgi isn't named for how it looks, it's named for the scientists who first described it and figured out what it was doing. The Golgi apparatus, their vesicles come from the endoplasmic reticulum and stick to this side. And again, here we see the use of the words cis and trans. We find these all over in science, not just in chemistry. So cis means close together, and trans means away from in science. So cis here, the cis face of the Golgi, it means close to the nucleus. The trans face is away from the nucleus. So that's how these terms are used here. So vesicles fuse with the cis face that are coming from the endoplasmic reticulum. They work their way through these pancakes up to the front, and then vesicles break off from the front edge. The pancake, once it reaches the front, breaks up into vesicles, and those vesicles are going to the outside of the cell. They're going to fuse with the surface of the cell and release their contents to the outside of the cell. So that's what the Golgi does. Molecules, as they're working their way through here, as the pancake they're in is working its way forward, changes are made. So the Golgi makes a few things, but mostly what it's doing is processing molecules and modifying them, adding sugar groups to things, cutting things, adding other modifications to proteins, and they're working their way to the surface. So the Golgi is the shipping center. These are things, everything that is secreted from a cell, whether it's hormones, neurotransmitters, extracellular matrix, cell wall components, all of it has to go through the Golgi to reach the outside of the cell. Lysosomes are made by the endoplasmic reticulum, and they contain enzymes that can digest cellular components. So that is why they need to be packaged in a vesicle. So the membrane vesicle keeps these enzymes that are in here from wreaking havoc in the rest of the cell and digesting general cell components. So for something to be digested by the enzymes in a lysosome, that vesicle has to fuse with another vesicle that contains the thing that's going to be digested. So if the cell is engulfing something, either as part of its function in a multicellular organism, or because it's a single-celled organism that eats things using vesicles, we call that process phagocytosis. So this cyto means cell, phago is eat, so phagocytosis is cell eating. So amoeba do this, they engulf their prey in little vesicles, and then a lysosome will fuse with that, and that's how they digest their food. Cells in our body do this. Cells that are part of the immune system, like macrophages do this, and natural killer cells do this. They fuse and engulf invading bacteria, infected cells that are our own cells that are infected with viruses. They engulf them and digest them by this same process of phagocytosis. Another function of the lysosomes is to digest things in the cell that are not functioning and need to be recycled. And this process is called autophagy or autophagy, self-eating. And this also uses lysosomes, and also another little organelle here, peroxisome, another vesicle that's produced by the endoplasmic reticulum. Like let's say there's a mitochondria that is no longer functioning. Perhaps a membrane is punctured or it's damaged in some way. It will be engulfed in a vesicle and digested by autophagy, by the process of autophagy. Peroxisomes are part of this process. Peroxisomes have many weird functions. They're smaller than lysosomes. They are necessary for breaking down long-chain fatty acids. They help oxidize toxins, and they also produce hydrogen peroxide as a byproduct. They both produce hydrogen peroxide and consume hydrogen peroxide, H2O2, from the cytoplasm. So that's where they get their name. Peroxisomes is that was one of the earliest functions of peroxisomes that was discovered. The protein enzymes are not always, but often seen as a crystal-like structure with a very regular structure. They have a few weird synthesis functions as well. Many of the functions of peroxisomes seem weirdly unrelated, and it's still being studied. There's still a lot of hypotheses about the peroxisomes that still require more data and testing. So we don't completely understand what the function of peroxisomes is, but they're essential for recycling, especially particular fatty acids, very long fatty acids, branching fatty acids, and also they're part of peroxide metabolism, both making it as a product of their oxidation functions and then breaking it down again. The mitochondria, which we'll talk more about, are very important in all eukaryotes. There are a few eukaryotes that have only very tiny and not useful mitochondria, but mitochondria are the only thing in our bodies that uses oxygen. Mitochondria and the fact their type of energy production, they make most of the ATP, over 90% of the ATP that your cells use, which is, remember, ATP is that little energy storage molecule. They make most of that using oxygen. So these are the only things in your body that use oxygen. There's nothing else in a human cell or a plant cell that needs oxygen. Nothing. Mitochondria are the reason that we need oxygen. That's why we need to breathe oxygen. So the reason that you die from lack of oxygen is because your mitochondria cannot make ATP anymore because you are not taking in new oxygen. Mitochondria have a double membrane, and it's the inner membrane that is the site of this ATP synthesis, making ATP from ADP using energy from our food. So, and we'll talk in a lot more detail about that when we get to respiration. Here's a nice TEM image of a mitochondria, and you can see that the inner membrane, all these little stripy looking things, those are folds in the inner membrane. So there's a single outer membrane and there's a single inner membrane, but the inner membrane has all these folds that go up and down in there, and you can see those in there. And it's on this inner membrane, stuck in that inner membrane, are all of the components that are making that ATP. And in the matrix inside here, that's where the Krebs cycle takes place, or the citric acid cycle, which is part of the process of breaking down our food. And again, we'll talk more about that later in the semester. Chloroplasts are another structure that has multiple membranes. The inner membranes, again here, which are colored green, are the site where photosynthesis occurs, or at least the photoreactions. That's where chlorophyll is. That's why these are colored green, is because that's where the green molecule, chlorophyll, is the green molecule that makes plants green. And that's where it's found, in these inner membranes. And these small little pancake-like membranes are called thylakoids. Don't worry about the word granum, but thylakoid is an important word for you to know. There are two outer membranes on chloroplasts, and then these thylakoids are membrane bound as well. And we'll talk a lot more about chloroplasts and photosynthesis later in the semester as well. So only eukaryotes have chloroplasts. Bacteria that are photosynthetic do not have chloroplasts. There are other types of plastids as well. So chloroplasts, that word chloro means green. Chloro is Greek for green. So chloroplasts are green plastids. So plastids are multi-membraned vesicles that we find in plants. We find them in many protists as well. And they can be green and photosynthesize. In red algae, they're reddish. And there are other kinds of plastids as well that are related to chloroplasts. And chloroplasts can turn into these as well, or turn back into chloroplasts, depending on what you do with the cells. Chromoplasts contain pigment and have other purposes, like for coloration of the plants. Leucoplasts, leuco means white. So leucoplasts generally are containing starch or are storing other compounds for the cell. And there's a few other kinds of plastids as well. And these are all related. So let's talk about the origin of eukaryotes and the origin of these plastids and mitochondria. So a hypothesis that had been around since the early 1900s was the hypothesis that chloroplasts primarily, and possibly even mitochondria, used to be free-living bacteria. Now, there were a handful of scientists that proposed this in the early 1900s, but they didn't have any evidence other than the fact that for chloroplasts had been seen under the microscope dividing in living plant cells. And that was very intriguing, but nobody ever really followed up on it. Later it was found that chloroplasts had, weirdly, DNA in them and were synthesizing DNA. That seemed peculiar because DNA was supposed to be in the nucleus. Well, in the 1960s, a researcher named Lynn Margulis promoted this hypothesis, and she was really the person who brought it all together and assembled the necessary proofs that, in fact, these people were right. And not only plastids like chloroplasts, but also mitochondria used to be free-living bacteria. And this hypothesis is called the Endosymbiont Theory. Symbiosis is two organisms living together. Endosymbiont is one organism living inside of the other. So her endosymbiont theory, her first observations to confirm this, were the fact that mitochondria and plastids, they just look like bacteria. Especially mitochondria are the same size as bacteria, 1 to 2 microns in size. Plastids tend to be a bit larger. And they divide, and she was using a transmission electron microscope, and she had many images like this where it looked like mitochondria were dividing. And then for chloroplasts, there was evidence from the early 1900s of living chloroplasts actually dividing with somebody observing them under the microscope. That was a report. So if they are the size and the shape of bacteria, and they kind of look like bacteria, and they divide, and the cells don't make them, nobody could find any place in the cell where mitochondria or chloroplasts seem to be being manufactured. For example, they weren't budding off of the endoplasmic reticulum. So they're dividing on their own. They kind of look like bacteria. So that was the hypothesis, that they once were free-living bacteria. This hypothesis leads to some predictions, as a good scientific hypothesis does, testable predictions. And one of the predictions would be, okay, if these were ancient bacteria that were engulfed by a eukaryotic membrane, we should expect the inner and outer membranes of mitochondria and chloroplasts to be different. We would expect the outer membrane to be like a eukaryotic cell, and we would expect the inner membrane to be more like a bacterial membrane. And that's in fact what was found, was that the inner mitochondrial membrane contains particular lipids that we would expect to find in bacterial membranes that are in fact very common in bacterial membranes. And those lipids make up a large portion of the inner membrane of the mitochondria, but not the outer membrane. And we also find that both membranes of the mitochondria contain proteins that we also expect to find in bacteria. So the membrane, especially the inner membrane of the mitochondria, is bacteria-like. It was also discovered that both mitochondria and chloroplasts have their own DNA chromosomes, and in fact it's a circular chromosome like a bacteria. They also have their own ribosomes and synthesize some of their own proteins. And remember that ribosomes was how Carl Woese distinguished bacteria, archaea, and eukaryotes. Well, the ribosomes that are inside mitochondria and plastids are in fact bacterial ribosomes. They are not like the eukaryotic ribosomes that are in the rest of the cell. We now know that in fact mitochondria and plastids only divide on their own. The cell cannot make them. If you remove all the mitochondria or plastids from a cell, the cell will never have any more. It cannot make them. These results have been repeated many times, not just by Lynn Margulis, but by many other researchers, and this is now a very accepted part of biology, that plastids and mitochondria were once free-living bacterial cells that were engulfed by an early eukaryote, very early in the history of life on Earth. So remember that I said that for over 1 billion years, the fossil record shows that there were only prokaryotes, and then suddenly these huge cells appear in the fossil record. So how did eukaryotes originate? So we have some distinguishing features here now. They're larger, they have internal membranes, and they have a nucleus. All eukaryotes have a nucleus. They also have mitochondria or remnants of mitochondria, and a subset, have chloroplasts. Not all of them have chloroplasts or plastids. So we can assume that chloroplast was a later event. So which came first though, mitochondria or the nucleus? That's a question. So scientists have been able to answer that question with DNA analysis. They can look at the genes that are involved with making the nuclear envelope and the ones that are involved with regulating the mitochondria, and ask which genes are older. And in fact, the nuclear envelope genes are older. So that indicates that the first thing that evolved in the eukaryotic lineage was the nucleus. So why did this evolve? Why did the nuclear envelope evolve? Probably for protection from viruses. One of the problems that any bacterial cell faces is constant attack by DNA viruses. So the nuclear envelope is excellent protection against DNA viruses. DNA cannot cross the nuclear membrane. It cannot get into the nucleus, and it can't get out of the nucleus. So it was probably protection from those DNA viruses, and it was an excellent protection from the DNA viruses. RNA is another story, and RNA can get in and out of the nucleus, including RNA viruses can get into the nucleus. But DNA cannot. So probably we have our ancient archaea here. Remember, eukaryotes evolved from archaea, with some foldings of the membrane. Those infold, and there are some archaea today that have some foldings of their membrane like this. At some point, these foldings evolved into a nuclear envelope and the endoplasmic reticulum, which are physically connected, and the nuclear envelope probably evolved from the endoplasmic reticulum. Then at some later point, a bacteria, which we now know the closest cousins of the mitochondria in the bacteria world are alpha proteobacteria. DNA analysis has indicated that as well. So some ancient alpha proteobacteria was engulfed by one of these early eukaryotic cells that had a nuclear envelope, but it had no ability to use oxygen to make energy. And it just kept this bacteria in its cytoplasm and allowed it to reproduce, and it provided a benefit to the bacteria of giving it a nice, safe place to live where it wouldn't be eaten, and the bacteria in return let the cell use all of the excess ATP that it was making. Some point later, one of these early eukaryotic cells engulfed a cyanobacteria, the only kind of bacteria whose photosynthesis produces oxygen as a byproduct. And so plants today, and photosynthetic protists as well, have both mitochondria and chloroplasts, and the rest of the eukaryotes have mitochondria or the disintegrated remnants of mitochondria and use oxygen to make ATP. So how did this endosymbiosis occur? Well, one of the ways that we can study it is by looking at more recent endosymbiotic events. So here's an example of that. This is a ciliate, it's a protist, and it engulfs another eukaryotic algae which live in its cytoplasm, and it's much smaller. This cell is much larger, about 150 to 200 micrometers in size, and the algae are 3 or 4 micrometers in size. So it engulfs these algae. They both benefit. The cell gets the products of photosynthesis, sugar, and the algae get a safe place to live, and they get supplied with minerals that this ciliate is taking in through its heterotrophic eating of things in its surroundings. So the fact that we can find situations today that are much more recent that are not obligate yet endosymbiosis. So in this example, we can separate these algae and continue to grow them independently, although we can't do that for chloroplasts. Here's another example with animals. Coral are animals, and coral take in algae from the water column, and those algae live inside the cells of the coral, but this relationship is, we call it not obligate, because we can separate them, and in fact this happens all the time. If coral becomes stressed, they expel all the algae, and the algae can survive in the water column, and the coral can survive for a while without their algae. This is called coral bleaching. And coral will do this in response to environmental stress like pollution or high temperatures. And eventually, if conditions improve and they think coral might be able to survive for maybe a year without their algae, the coral can take algae in again if it's still alive and go back to being having the benefit of having a photosynthetic organism living inside. So this is, again, another situation where the relationship is not completely obligate yet because we can separate them, but it's a more recent endosymbiosis. So the cyanobacteria endosymbiosis that occurred that led to chloroplasts just happened much longer ago. So long ago that the chloroplast can no longer live independently. So DNA analysis has given us a lot of new information about this. When scientists sequenced the DNA, the little chromosomes that are found in mitochondria, and did a phylogenetic tree of that DNA, and they asked, did this event happen multiple times, or are all mitochondria descended from some first cell? And it looks like mitochondria are each other's closest relatives, that whatever bacteria became the endosymbiont is probably now extinct because we can't find a super close relative of the mitochondria. But there are many cousins in the alpha proteobacteria. Alpha proteobacteria, many of them currently live as internal parasites inside other cells, and they can be expelled, so it's not an obligate relationship. So it seems that this is very, very likely. So what that means is that all eukaryotes alive today, protists, plants, fungi, animals, are all descended from one cell that engulfed an alpha proteobacteria. And that alpha proteobacteria is the ancestor of all the mitochondria in all the eukaryotic cells today. So that's pretty cool. I think this is one of the coolest discoveries in biology in the last 200 years, is the discovery of that mitochondria and chloroplasts were once free-living bacteria. And there was a lot of pushback on it at first, because, you know, when you have a hypothesis that is an extraordinary claim, you need a lot of evidence to back it up, to convince people. And it took Lynn Margulis over 10 years with piles and piles of evidence to convince everybody. So initially for chloroplasts, it was thought that perhaps there were multiple origins, because there's red algae and green algae, and it was thought, well, maybe they're not really related, and there's golden algae as well. But it turns out when they do the same thing, when they take the little DNA that's in these chloroplasts and any other kind of plastid, and they compare the sequences, the same thing is true. There's a single origin for all plastids. And their closest cousins are cyanobacteria, which cyanobacteria are the bacteria that evolved a new kind of photosynthesis that introduced oxygen, free oxygen, into the sea and the air of Earth for the first time. So the different lineages that we see, like green algae versus red algae, those differences evolved later, after the origin of chloroplasts. So what is all the evidence now for endosymbiosis? There's a few more things that I haven't put on the list, but these are the important ones. The first thing is that mitochondria and chloroplasts divide on their own. Cells don't make them. They can divide when the cell isn't dividing. So for example, if you embark on an exercise program and start increasing your aerobic fitness, one of the things that happens is your mitochondria will increase in number in your muscle cells and in your heart muscle. So mitochondria divide on their own, and so do plastids. They have their own circular chromosomes, and when we analyze the genes on those chromosomes, those genes are bacterial genes. They are not genes that we would typically associate with the eukaryotic nucleus. Their ribosomes are bacterial ribosomes, and they make some of their own protein. And I say some because they've lost a lot of their genes. So a typical bacteria has 3,000 to 4,000 genes, and mitochondria have less than 20, at least for human mitochondria. Human mitochondria have fewer than 20 genes left on their little circular chromosomes, but they still make some of their own gene products with their own little bacterial ribosomes. And the rest of their needs are supplied by the cell, which is why they can't live independently anymore. They are surrounded by a double membrane. The inner membrane of mitochondria is like a bacterial membrane, and that is true as well for chloroplasts. Those thylakoid membranes where photosynthesis is actually taking place, those membranes are more like a cyanobacteria than they are like a eukaryotic cell. But these events occurred so long ago that although many people have tried, no one has been successful culturing mitochondria or chloroplasts on their own like a cell. So they're not true cells anymore because they can't live independently. They are totally dependent on the cells that they live in for proteins and other gene products. So that's the end of this section. And we will continue in one more section with the rest of cell structures.